Good day, sir. So today I'm going to discuss a research article related to muscle physiology. The research is entitled Mitophagy Directs Muscle-Adipose Crosstalk to Alleviate Dietary Obesity. The researchers are Tingting and others. This belonged to the journal called Cell Reports, published at Science Direct on May 1, 2018. For a quick insight about this research, it mainly focuses on the role of mitophagy in regulating metabolic disorders such as obesity. So, while mitochondrion is known as the organelle that fuels biochemical processes in the cell, it is also important for us to take note that it collectively maintains homeostasis in conjunction with the skeletal muscle. On the other hand, mitophagy serves as a major quality control mechanism for selective targeting and removal of damaged or dysfunctional mitochondria. Mitophagy actually originated from two words. We have mitochondria and autophagy. As we all know, autophagy is a cellular mechanism that functions to remove damaged cells. On the other hand, we have mitochondria, and as mentioned earlier, the powerhouse of the cell. Taking these two concepts together, we come up with the word mitophagy. So this mechanism is similar to autophagy, only mitophagy is specific to mitochondria alone. So the dysregulation of mitophagy has been implicated in the pathogenesis of many chronic diseases related to metabolic disorders. Hence, this research targets to identify the connection and role of mitochondrial quality control in skeletal muscle for maintaining metabolic homeostasis during adaptive stress response. Meaning, this research hopes to provide evidences that identifies or clear out the main role of mitophagy in maintaining metabolic homeostasis. So let's proceed to methodology. This is the overview of the entire processes conducted by the researcher. They utilized two models, which is the FANC1 MKO or FANC1 knockout mice model and the wild-type mice as the control. They were subjected to several tests and evaluations. So FANC1 MKO mice was produced by breeding FANC1 flox mice with myosin creatine kinase and born at normal Mendelian ratios, and they were grossly normal on inspection. By the way, FANC1 is a regulatory gene of mitophagy. So in this model, FANC1 was totally deleted. So these models were subjected to several tests. So let's proceed to the first part. You have here the evaluation of muscle fuel utilization during exercise. So the mice models were subjected to high intensity exercise protocol, and the RER and VO2 were measured. The results confirmed that there was a higher real-time respiratory exchange ratio in FANC1 relative to wild-type model, and it was also confirmed that there was less oxygen utilization and reduced maximal peak oxygen consumption among the models. This only suggests that they consume less fat and depend on glucose metabolism. For further assessment of physical impacts of impaired muscle fat utilization, the mice models were subjected to low intensity endurance exercise. So since endurance exercise performance depends heavily on fat utilization, the researchers hypothesized that muscle fund C1 deficiency would worsen exercise intolerance on an endurance exercise regimen. So taking all of those together, it implies that the skeletal muscle specific deletion of FANC1 causes decreased muscle fat utilization and endurance during exercise, meaning the total loss or removal of FANC1 resulted to lower fat utilization. Let's proceed to the next part. We have here defining molecular basis of the reduced muscle fat utilization and endurance during exercise using biochemical hallmarks of mitophagy. So the LC3, 2, and P62 protein levels were markedly reduced this time. So autophagy flux analysis was used by the researchers to confirm impaired mitophagy. The mice were treated with colchicine. So the um, The results suggest that an LC3-mediated mitophagy defect in FANC1 MKO muscle. So, taking the results together, it implies that in vitro manipulation of FANC1 did recapitulate defective mitophagy and impaired mitochondrial energetics as observed in FANC1 MKO muscle, meaning after the specific deletion of FANC1 in the mice model, it portrayed or it exhibited impaired mitophagy. So let's proceed to the third part. We have here the evaluation of metabolic stress by feeding normal chow diet and high-fat diet. This time, the mice models were subjected to two different diets. So in normal chow diet, it showed slight decrease in energy expenditure at night with similar metabolic phenotypes in FANC1 MKO mice relative to the wild-type mice. The results in high-fat diet, on the other hand, suggest that loss of muscle FANC1 alleviates HFD-induced obesity. So this is contrary to the hypothesis mentioned earlier. This time, the researchers highlighted that deficiency of FANC1 is actually optimal to metabolic homeostasis, unlike the total removal of FANC1. So the determination of glucose homeostasis also suggests that lower glucose and insulin levels um, impose sensitivity to insulin. So taking these all together, muscle FANC1 deficiency actually protects against HFD-induced obesity and improves glucose homeostasis, unlike the total removal of it from the muscle. So let's proceed to the third part. We have here mitochondrial remodeling in skeletal muscle for further insights about skeletal muscle energy metabolism. So multiple lines of evidence confirm a decreased mitochondrial energetics in HFD-fed FANC1 MKO muscle. There was also a reduced ATP production and significantly lower respiration rates. Testing whether muscle FANC1 deficiency can trigger thermogenesis through FGF21 dependent muscle-adipose crosstalk. So after the test, the results provide strong evidence that FGF21 is required for the observed muscle FANC1 deficiency mediated favorable effects on systemic metabolism. So during the conduct of the, I mean uh, during the in the conduct of tests where the mice models were subjected to high-fat diet, there was an increase in FGF21. So um, this substance contributed to the FANC1 deficiency mediated favorable effects in regulating the metabolic homeostasis. So taking all these together, muscle FANC1 deficiency leads to activation of adipose tissue adaptive thermogenesis through endocrine actions of FGF21. So what is thermogenesis? This is a temperature increase in the body. So meaning during the deficiency of FANC1, there was also an increase in temperature among the mice models. Okay, so in conclusion, we have here skeletal muscle specific deletion of FANC1 causes decreased muscle fat utilization and endurance during exercise. The loss of FANC1 impairs mitochondrial energetics in skeletal muscle. Muscle FANC1 deficiency protects against HFD-induced obesity and improves glucose homeostasis. I would like to point out that the total deletion of FANC1 resulted to um, decreased fat utilization, and FANC1 deficiency actually protects against HFD obesity and rather improves glucose homeostasis. So muscle FANC1 deficiency leads to activation of adipose tissue adaptive thermogenesis through endocrine actions of FGF21. So this study has revealed a crucial role of FANC1-dependent mitophagy in mediating the muscle-adipose dialogue that regulates systemic metabolism. In other words, the researchers were able to identify that FANC1 actually plays a major role in regulating homeostasis in the body in concert with mitophagy, since it is a regulatory gene of the mitophagy. So, meaning FANC1 is really an important regulator of muscle fat utilization and endurance capacity during exercise. The results suggest that muscle mitophagy pathways directs muscle FANC1 deficiency to trigger thermogenesis or temperature increase through FGF21 dependent muscle adipose crosstalk that alleviates metabolic disorders such as obesity. So, as we can see here in this illustration, the total removal of FANC1 resulted to the dependence of the mice models in utilizing ATP, meaning they are dependent in glucose metabolism. And as for the FANC1 deficiency, it initiated adaptive thermogenesis where the glucose metabolism was regulated and the use of adipose tissues were regulated. So for the self-assessment, I have realized that this is actually an extensive research that utilized several techniques to narrow down factors that influence the nature of mitophagy in skeletal muscle. So earlier, there were incorrect assumptions, but the researchers were able to correct them in later tests, which is actually nice because it gave a clearer view about the study. So in relation to metabolic disorders, this research highlights not only significant factors for social implications, but also this research contributed in regulating overall health and wellness of an individual, since um, obesity is closely associated to medical complications. With all that being said, this research added a pivotal contribution to the existing body of knowledge in the field of disease management, health, and medicine. So I guess that's all. Thank you, sir.